maybe next week. We'll see. But I'm going to go ahead and jump into this. So I'll start with number 1. What I do for these is, I was taught in another class, we have 4 different traits in this one, or 4 different letters. And so all I do is I write it out. I don't know if you guys can see this. I just do a bunch of monohybrid crosses. And then from there, I write the percentages of whether, what the percentages for homozygous dominant, heterozygous, or homozygous recessive. And it'll always be, half or a quarter in this case. And then just depending on whatever the question asks. So like A is asking, for a certain genotype, and I got 1 in 128. I just multiply across based on what it's asking. And you do the same thing for B. I get 1 over 256. C is 0 because there's no homozygous recessive D trait. And then D is 1 in 32. So that's basically all you have to do for number 1. And then number 2, I'm just going to go ahead and try and read through this pretty quickly. But it's just paired homologous chromosomes are sometimes called bivalent or tetrads. B, the structure that connects the centromere to the microtubules of the meiotic spindle is called a kinetochore. And then C is a diploid fruit fly, has 4 pairs of chromosomes. So a fruit fly cell in prophase 1 has 16 chromatids, where a fruit fly cell in metaphase 2 has 8 sister chromatids. And then for D, a test cross is a type of genetic cross. I can determine if a purple flower pea plant has a homozygous dominant or heterozygous genotype. The reason I go with test cross is just, that's how you can find out what the paternal and maternal genotypes were. E, a non-functional allele, is a null allele, and a partially functional allele is called a hypomorphic allele. And then if I move down to F, I got here Mendel's law of independent assortment involves 2 traits, while his law of segregation involves just a single trait. And then for number 3, it asks you to diagram all possible homologous chromosome arrangements. And so there's basically, I guess, 4 different arrangements. And we know that in every single one, based on the genotype provided, is that it's going to be homozygous dominant for the T trait. So you're going to have capital TT for every single one. And you just do all the different combinations between the R's and the Y's, switching them off between dominant and recessive so that you get the heterozygous in there. And so that's how they arrange themselves. For B, it has to indicate all possible gamete genotypes, and so there's 4 of them. So you have capital T, capital R, capital Y, and you have capital T, and then you have recessive for R and Y, and then you have capital T, lowercase r, capital Y, and then you have capital T, capital R, lowercase y. So once again, you're just kind of flipping the R's and Y's. And then C and D, sister chromatids, there's going to be 12. For D, there's going to be 3 chromosomes during metaphase 2. And it's pretty self-explanatory. For number 4, it says a horse has 32 pairs of chromosomes. So how many are present per horse cell at the conclusion of meiosis 1? It's 32. How many tetrads are present during prophase 1 of meiosis? It's 32 as well. How many sister chromatids are present in a horse cell during prophase 1 of meiosis? Then we have 128. We're doubling 64. And then D, how many sister chromatids are present per horse cell during metaphase 2 of meiosis? So 64. And then E, how many bivalents are present per horse cell during anaphase 2? And that's 0. And then, last but not least, if we move down to number 5, we're once again just doing, we have 4 different genes. And so it's a tetrahybrid cross, and all we're doing is a bunch of monohybrid crosses, just like number 1, and then we do the percentages the same way. And so I have another piece of paper just like that. So for A, it asks which will be tall, round, green, and terminal. That's 9 out of 64, so 14%. And then B gives you a certain genotype, and it'll end up being 1 in 32. C, what proportion of the offspring will be heterozygous for all 4? And that's 1 in 16. It's just half times half times half times half. And then D, what proportion of the offspring will be dwarf, wrinkled, yellow, and axial? So that's 1 in 64. But that's me just getting through the homework there.